Dear friends, plant breeding ഏറ്റവും പ്രധാനപ്പെട്ട ഒരു ടോപ്പിക്കാണ് നമ്മൾ ഇന്ന് ഡിസ്കസ് ചെയ്യാൻ പോകുന്നത്. The hybridization and the hybrid Vigour. ഒരുപക്ഷെ നമ്മൾ പ്ലാന്റ് ബ്രീഡിംഗ് എന്ന് കേൾക്കുമ്പോൾ ആദ്യം ചിന്തിക്കുന്ന പ്രോസസ്സ് ഹൈബ്രിഡൈസേഷൻ തന്നെയാണ്. എന്താണ് ഹൈബ്രിഡൈസേഷൻ, എന്തിനു വേണ്ടിയാണ് ഹൈബ്രിഡൈസേഷൻ നടത്തുന്നത് എന്നതിനെപ്പറ്റിയൊക്കെ പ്രാഥമികമായ ധാരണ നമുക്കുണ്ട്. ഇന്ന് നമ്മൾ വിശദമായിട്ട് എങ്ങനെയാണ് ഹൈബ്രിഡൈസേഷൻ്റെ പ്രോസസ്സ്, എന്താണ് ഹൈബ്രിഡൈസേഷൻ്റെ പിന്നിലുള്ള അടിസ്ഥാനം, പിന്നെ ഹൈബ്രിഡൈസേഷൻ്റെ തുടർച്ചയായിട്ട് കാണപ്പെടുന്ന ഒരു പ്രധാനപ്പെട്ട പ്രതിഭാസമാണ് ഹൈബ്രിഡ് വിഗർ അഥവാ ഹെറ്ററോസിസ് എന്ന് പറയുന്നത്. എന്താണ് ഈ ഹൈബ്രിഡ് വിഗർ തുടങ്ങിയ കാര്യങ്ങൾ നമുക്കൊന്ന് കാണാം. Let us move on to the first slide. What is meant by hybridization? ഈ രണ്ട് പാരഗ്രാഫിലായിട്ട് തന്നിരിക്കുന്നത് ഹൈബ്രിഡൈസേഷനെ രണ്ട് തരത്തിൽ ഡിഫൈൻ ചെയ്യുന്നതാണ്. ഒന്ന്, എന്താണ് ഹൈബ്രിഡൈസ്ഡ് പ്രോഡക്റ്റ് അല്ലെങ്കിൽ വാട്ട് ഈസ് എ ഹൈബ്രിഡ് എന്നതിൻ്റെ ഡെഫിനിഷനിൽ നിന്ന് ഹൈബ്രിഡൈസേഷൻ പ്രോസസ്സ് എക്സ്പ്ലെയിൻ ചെയ്തിരിക്കുന്നു. അങ്ങനെ പറയുകയാണെങ്കിൽ നമുക്ക് പറയാം, ഇൻഡിവിജ്വൽ പ്രൊഡ്യൂസ്ഡ് ആസ് എ റിസൾട്ട് ഓഫ് ക്രോസ് ബിറ്റ്വീൻ ടു ജനറ്റിക്കലി ഡിഫറൻറ്റ് പേരൻസ് ഈസ് നോൺ ആസ് എ ഹൈബ്രിഡ്. വ്യത്യസ്തമായ ജനിതക ഘടനയുള്ള രണ്ട് ഓർഗാനിസംസിനെ തമ്മിൽ, രണ്ട് ജീവികളെ തമ്മിൽ ക്രോസ് ചെയ്ത് നമ്മൾ ഒരു പുതിയ പ്രോജനി ഉണ്ടാക്കുകയാണെങ്കിൽ അതിനെ നമുക്ക് ഹൈബ്രിഡ് എന്ന് വിളിക്കാം. ദ നാച്ചുറൽ ഓർ ആർട്ടിഫിഷ്യൽ പ്രോസസ് ദാറ്റ് റിസൾട്ട് ഇൻ ദ ഫോർമേഷൻ ഓഫ് എ ഹൈബ്രിഡ് ഈസ് നോൺ ആസ് ഹൈബ്രിഡൈസേഷൻ. അങ്ങനെ ഒരു ഹൈബ്രിഡിനെ ഉണ്ടാക്കുന്ന പ്രവൃത്തിയെ, അത് നാച്ചുറൽ ആവാം അല്ലെങ്കിൽ ആർട്ടിഫിഷ്യൽ ആവാം, ആ പ്രവൃത്തിയെ നമ്മൾ ഹൈബ്രിഡൈസേഷൻ എന്ന് പറയുന്നു. മറ്റു തരത്തിൽ, ദ പ്രൊഡക്ഷൻ ഓഫ് ഹൈബ്രിഡ് by crossing two individuals of unlike genetical constitution, that means non-similar genetical constitution, entirely different എന്ന് പറയാൻ പറ്റത്തില്ല, difference, the genetic constitution having much difference is known as hybridization. Hybridization is an important method of combining characters of different plants. This is the most important thing. We can combine the characters of organisms with the process of hybridization. It doesn't change the genetic content or genetic makeup, but it is only a mechanism for the recombination. It produces the new combinations of genes in an organism. That is the most important thing about hybridization, and that is the major difference between mutation and hybridization. In hybridization, no change in genetic material or DNA is involved, but it is only a recombination mechanism. It is an interaction. Here the objective is the interaction between the alleles. So the first natural hybridization was recorded by Cotton Mather in 1716. So it is a very ancient mechanism, it is method. It can be considered as an art also. That's why we say the plant breeding is an art, because before knowing the basic principles of sexual reproduction or sexual recombination, people started working with hybridization. And the work was in corn in 1716. The first artificial interspecific plant hybrid was produced by Thomas Fairchild in 1717. So it is also very ancient, and it was called as Fairchild mule, because as we know, the mule is a interspecific cross. Here the first interspecific plant was a hybrid. The plant hybrid was produced and it was nicknamed as Fairchild mule. Hybridization was first of all practically utilized in crop improvement by Joseph Colruyter in 1760. But after Mendel, the hybridization had become the key method for crop improvement. Today, it is the most important common method for crop improvement or plant improvement, and the vast majority of crop varieties have been produced by this method. So what are the objectives of hybridization? The first one is to artificially create a variable population for the selection of types with desirable traits or desirable combinations of characters. Second one is to combine desired characters into a single individual. That means if two characters are seen in two different varieties, two desirable traits are seen, and we want to combine them in an individual, we try to hybridize them. Similarly, the third objective is to exploit and utilize the hybrid varieties. So there are different types of hybridization based on the parents used. The first one is the hybridization between varieties, which is known as the intra-varietal hybridization. And that means the crosses are made between the plants of the same variety. Within the variety, there are much variations. To utilize the new combination of such varieties or such variations, we perform intra-varietal hybridization, and it is very simple and easy. Then comes the intervarietal or intraspecific hybridization. That means the hybridization is between two different varieties belonging to a particular species. And there is interspecific hybridization or intrageneric hybridization, where we use two different plants belonging to two different species of the same genera. Similarly, there is an introgressive hybridization. That means when we want to transfer a particular character, a specific character into a normal variety, we use introgressive hybridization, and it is aimed at the transfer of some genes from one species into the genome of other species, and it is said to be introgressive. Then there is another term for the crosses above the level of species. If it is within the species or intervarietal hybridization, we call it as close hybridization. But when it is above the rank of species, such as interspecific, we call it as distant hybridization. The cross between different species of the same genus or different genera of the same family are known as the distant hybridization or wide crossing. Such crosses are called distant crosses. It is of rare occurrence, and the chance for success is also rare. So what is the procedure of hybridization? കുറച്ച് വേഗത്തിൽ നമ്മൾ പറഞ്ഞു പോവുകയാണ്. നിങ്ങൾക്ക് അറിയാവുന്ന കാര്യങ്ങൾ തന്നെയാണ്, പക്ഷേ നമ്മൾ സ്വല്പം വേഗത്തിൽ ഈ പ്രോസസ്സ് പറഞ്ഞു പോവുകയാണ്. പരീക്ഷയ്ക്ക് ചോദിക്കാവുന്ന ഒരു ക്വസ്റ്റ്യനാണ് ദ പ്രോസസ് ഓഫ് ഹൈബ്രിഡൈസേഷൻ, ഓർ വാട്ട് ആർ ദ തിങ്സ് ദാറ്റ് ഷുഡ് ബി ടേക്കൻ കെയർ ഓഫ് ഡ്യൂറിങ് hybridization process, or how will you plan for a hybridization? What are the steps involved in hybridization or artificial hybridization? All these are questions asked. So be careful and understand, but it's very simple. The major steps are the selection of the parents. We have to select the parents, then selfing the parents. For the purpose is to make the parents homozygous. The parents we select may have the characters of hybrid. The alleles may be different, but we try to self-fertilize them by artificial self-pollination to make the genes homozygous. Then the emasculation. Emasculation is the removal of male sex organs from the female parent. Then the bagging. It is protected from other pollens that may come as a part of natural pollination. So we avoid this kind of normal pollination by bagging them. Then the tagging the parents to identify the stage of hybridization, and the process of crossing, the artificial pollination. We take the pollen from the desired male parent and pollinate on the female parent. Then the harvesting of seeds and storing of F1 seeds. The seeds produced in after pollination is considered as F1 seeds, and the seeds germinate to give rise to the F1 progenies. So collect the seeds from the fertilized flowers, then raising the F1 generation. These are the steps involved in the process of hybridization. We will see all these steps in detail. All these are simple steps, but you have to write something about all these steps. The selection of parents. So the selection of parents depends upon the aim and objectives of the breeding. Parented plants must be selected from the local areas and are supposed to be the best suited to the existing conditions. Would we cross only the best individuals? Because our objective is to raise a superior variety. So we select the parents very carefully, then selfing the parents. It is essential for inducing homozygosity, as I said earlier, or eliminating the undesirable character combinations and obtaining inbreeds. Then the emasculation, the third step is emasculation. What is emasculation? It may be asked as a short question. It is the third step in hybridization. Inbreeds are grown under normal conditions and are emasculated. It is the removal of stamens from the female parent before they burst and shed their pollen grains. So, before the anthesis of the flowers are artificially opened and the anthers are removed from the flower so that the release of pollen grains in the vicinity of the stigma is avoided. It can be defined as the removal of stamens or anthers or the killing of pollen grains of a flower without affecting in any way the female reproductive structures or reproductive organs. Emasculation is not required if the plant is unisexual, but it is essential in bisexual or self-pollinated crops. Then what are the methods for emasculation? It is also a very important thing. How can we emasculate it? One is hand emasculation or forceps or scissor method, where this method is generally used in plants which have large flowers that we can mechanically remove the anthers. In this method, the corolla of the selected flowers is opened and the anthers carefully removed with the help of a forceps or scissors. So, there are much precautions to be taken before the hand emasculation. The flowers should be selected at a proper stage. It should be too mature or too immature. If it is at the correct stage, the developmental of the flower may affect it. If it is too early, the female reproductive organs may not develop properly. If it is too late, pollen grains may have shed into the pollen, so our hybridization experiment may fail. So, the time of emasculation is very important. Then the stigma should be receptive and anthers should not be dehisced. This is the condition. If in the case of it is when the stigma become receptive, usually it is the time of anthesis, the stigma become most receptive. So, the stigma should be receptive for our pollination process and the anthers should be removed before the anthers are shed or the pollen grains are shed. All the anthers should be removed from the flowers without breaking. So, without releasing the pollen grains, we should remove the anthers very carefully. Stigma and ovary of the flower should not be damaged. That is a very important thing because we have to preserve the stigma for the actual hybridization process or pollination process. In the figure, we can see the removal of anthers using a forceps. Then the hot water treatment, it is the removal of stamens with the help of a forceps is difficult in small plants, small flowers. In such cases, the hermaphrodite flowers or bisexual flowers are emasculated by dipping the flowers in hot water for 1 to 10 minutes. The time may vary species to species, but this method is based on the fact that Gynoecia can withstand the hot temperature at which the anthers are killed. In this method, an equipment is used which is placed in a simple heavy stand. We can see the picture of that mechanism. It consists of a cylindrical metallic container of 2 feet length with one hole of 5 cm to 16 cm diameter, and it is based on the size of the inflorescence we use. After inserting the panicle inside the container, a cork is fitted on the hole to close it. A 35 cm long rubber tube or belt is stretched over the side of the container, and when in use, this tube tied around the peduncle of the head. To measure the temperature, in the upper side of the container, a thermometer is placed. In the field, water is carried in a jug or a flask. Then, this is the mechanism. The panicle is inserted into the container prior to blooming for a particular duration of time. It has been observed that pollen grains of rice are killed by immersing the inflorescence for 5 to 10 minutes in 40 to 44 degrees Celsius in a thermoflask. This is the setup. As we said, we can keep the inflorescence into this chamber and tie it here, and this mechanical container contains the water. So, the thermometer is placed in this here, to measure the temperature, and by this mechanism, by keeping the inflorescence in the hot water for 10 minutes, we can kill the anthers. Then the cold water treatment, like hot water treatment, cold water also kills pollen grains without damaging the gynoecium. In rice, 0 to 6 degrees Celsius temperature is maintained to kill the pollen grains. This method is less effective than hot water treatment because most of the plants can withstand lower temperature. Then the alcohol treatment, method is also not commonly used because the duration of treatment should be very less. Very short duration only we can expose the plant to the alcohol. Flowers or inflorescence are immersed in alcohol of a suitable concentration for a brief period, usually 70 to 80% alcohol is used. In alfalfa, a treatment of even 10 seconds with 57% alcohol is sufficient. That means it is the lower limit. So 10 seconds is very precise duration, so it is not used commonly. Then suction method in mechanical method and is suitable for crops having minute flowers. In this method, the amount of pressure is applied in such a way that only anthers are sucked out and the parts of flowers like gynoecium remain intact. However, in this method, 10 to 15% self-pollination chance is there because it is the air that suck the anthers. So the anthers may burst and the pollen grains may release. So it is one of the major drawbacks, but it can be done in bulk. We can suck the anthers of flowers which are growing in a field very easily. Then male sterility is the most important method we used. It is the emasculation option can be eliminated by the use of male sterile plants. In some self-pollinated plants, for example, such as sorghum, onion, barley, etc., anthers are sterile and do not produce any viable pollens. Self-incompatibility is also another method where the self-pollination is prevented in such species. We have studied those things in biotechnology where some mitochondrial sequences are important or related with the cytoplasmic male sterility. So it is a common thing which is employed in modern days for the emasculation. It is not a method of emasculation, it is a mechanism to employ the it is a mechanism employed instead of the emasculation process. Then the chemical gametocides. Certain chemicals are capable of causing male sterility when sprayed before flowering, such as 2,4-D, NAA, malic anhydrosine, tribenzoic acid, etc. All these are different types of plant hormones. So in cotton, which is done in large quantities, this method is used for emasculation, where these chemicals prevent the development of male sex organs. So these are the different methods for emasculation. After emasculation, we do the bagging. Bagging means it is the fourth step in the hybridization process after emasculation. The emasculated flowers or inflorescence is immediately bagged in paper covers to avoid the entry of any foreign pollen. The bags may be made of paper, butter paper, glassine or fine cloth. Butter paper vegetable parchment bags are the most commonly used. The bags are tied to the base of the inflorescence or to the stalk of the flower with the help of thread, wire or pins. The bagging is done with the emasculation in bisexual plants and before the stigma receptivity and dehiscence of the anther in unisexual plants. Both male and female flowers are bagged separately to prevent contamination in male flowers and cross-pollination in female flowers. That means even if there is a cytoplasmic male sterility or some other mechanism, or the flowers are unisexual, we have to bag it because cross-pollination may occur. The pollen grains may come from other plants. So we have to bag it even though a proper emasculation is not required in the case of a unisexual plant. So this is the kind of bagging. Bagging can be done for inflorescence or the entire panicle or the entire terminal region of the plant, and a tag is added to the bagged plant. So tagging, the emasculated flowers are tagged just after bagging. Generally, circular tracks of about 3 cm or rectangular tracks of up 3 into 2 cm are used. I don't think that is not important because this is the standard size used in breeding institutes. So you have to mention it wherever required. The tags are attached to the base of the flower or inflorescence with the help of a thread. So this is the bagging. The information on the bag, it is also very important. Don't think that it is just a paper with some writings. The following things should be there in the tag. The number referring to the field record. That means we maintain a field record in a book in our lab. So there should be a number for the work we are following. So it should contain the number. Then the date of emasculation should be there. Date of crossing should be there. At the time of emasculation and bagging, we have to keep space for entering the date of hybridization or date of crossing. Then the name of female parent is written first, followed by a cross sign, and then the male parent. For example, C into D means C is the female parent and D is the male parent. So we can remember now there are two varieties of coconut plants in Kerala, T into D and D into T. T means tall variety crossed with dwarf variety, where tall is the female and dwarf is the male. And D into T, where D dwarf is the female and tall is the male parent. So next is the crossing. It is the sixth step. It can be defined as the artificial cross-pollination between the genetically unlike plants. In this method, mature, fertile and viable pollens from the male parent are placed on the receptive stigma of the emasculated flowers to bring about fertilization. We transfer the pollen grains from the desired male parent and place it on the surface of the stigma of the male parent. Pollen grains are collected in petri dishes in the case of cotton or wheat, or in paper bags, for example in the case of maize, and applied to the receptive stigma with the help of a brush or with a piece of paper, toothpick or forceps. It is based on the experience, trial and error method. We find out the most suitable material for that particular pollen grain. In some crops, the inflorescence of both the parents are enclosed in the same bag. This is also done. For example, in the case of sugarcane also this method is used. The male parent and female parent are grown side by side, and at the time of hybridization or at the time of crossing, we put both the inflorescences under a single cover so that pollination takes place. Then the harvesting and storing of F1 seeds. Crossed heads or pods desirable plants are harvested, and after complete drying, they are threshed. Then the seeds are stored for sowing with proper tagging. Then the F1 generation is grown in the coming season, and the stored seeds are sown separately to raise the plants. The plants of F1 generation and progenies of cross seeds, and therefore they are hybrid, and proper data should be collected from about their characters so that we can analyze the success of hybridization. So these are all about the process of hybridization. Next we move on to the next topic, that is the hybrid vigor. Hybrid vigor is one of the important objectives of hybridization. Hybrid vigor is nothing but the more vigorous nature of the F1 hybrids. When two homozygous inbreeds, that means a true breeding line obtained by continuous inbreeding of genetically unlike constituents are crossed together, the resulting hybrids obtained from the crossed seeds are usually robust, vigorous, productive and taller than either of the parents. So you should remember this definition as such. It is nothing but the F1 hybrid is more vigorous than the parent, but you should write all these things because these are the characters in which the plant show its vigorousness. This increased productivity or superiority over the parents of the F1 is known as the hybrid vigor or heterosis. Heterosis can be defined as the superiority of the F1 hybrid over both parents in terms of yield or some other characters. So heterosis has been known since the art of hybridization, as I said in the last stages of 16th century. Karl Reuther was the first to report hybrid vigor in the hybrid of tobacco. Datura, etc. Mendel in 1865 observed this in P crosses. That means the F1s were more vigorous than their both the parents. Darwin also reported that inbreeding in plants results in the deterioration of vigor, and the crossing result in the hybrid vigor. That means if plants are continuously inbreed or self-pollinated, then the vigor decrease. If we promote outcrossing, distant hybridization, the vigor increases. On the basis of his experimental, Bale concluded that F1 hybrids yield as much as 40% more of the parental varieties. It is a overall estimate on the yield, not based on the individual reasons for the increase, but it is noted that in a F1 hybrids around 40% of increase in yield was noticed. From subsequent studies, it was found that intervarietal crosses in maize also showed the same results. While discussing the work of maize during 1914, it was G.H. Shull proposed the term heterosis. Hetero means of different, osis means condition. That means the difference bring about a much higher yield. That's the meaning of this. Means the meaning of this word. Poweri reported that the crossing, however, may result in either weak or vigorous hybrids as compared to parental hybrids. That was another observation, that the progeny sometimes may become weaker, or most of the time it become more vigorous. So these are the historical account on the hybrid vigor. The hybrid vigor is synonymous to heterosis. They denotes the same thing. It is generally agreed that hybrid vigor describes only superiority of the hybrid. But we cannot say that the change in the hybrid. So heterosis means it is the superiority of the hybrid over the parents, while heterosis describes other situation as well. That's a difference in the usage of these things. Hybrid vigor means it is positive, it is vigorous, but heterosis may be a difference, that may be a positive difference or negative difference. That is the difference in the usage. Many hybrids in the tomato are early maturing ones in the case of F1 hybrids. Then types of heterosis. There are two types of heterosis. One is true heterosis and the other is pseudo heterosis. True heterosis means it's a sustainable type of change. That means it exists as a change in the organism, but in pseudo heterosis, crossing of the two individuals gives a temporary change in the performance. It is an accidental or excessive or unadaptable expression of the temporary vigor and the vigorous overgrowth. It is called as pseudo heterosis. But in the case of true heterosis, it is clear that the progeny is superior to the parents sustainably. And there are two mechanisms are proposed for this true heterosis. One is mutational true heterosis. It is that it is the sheltering or shadowing or masking the unwanted or deleterious alleles in the F1. Recessive mutant genes by their adaptively superior dominant alleles, so that the recessive alleles are not expressed and the organism shows a superiority. And the balance to true heterosis, it means that it is like the cis transmutations, like where both the kind of mutations, both in the parents, contribute to superiority. It arises out of balanced gene combinations. In one parent, one allele will be better and the other allele will be better in the other parent. So when these alleles come together, they give an adaptive balanced gene combination, so that true heterosis is observed. Then what are the causes of heterosis? The phenomenon of heterosis can be explained on different genetic processes. We mentioned the genetic causes are the dominant hypothesis and over-dominance hypothesis. The dominance hypothesis was proposed by Davenport in 1910, Bruce 1910, Cable and Pellow in 1910. So all these are earlier theories without knowing the basic principles of genetics. or DNA, but it is assumed that the hybrid vigor result from the bringing together of female dominant genes. According to this theory, genes that are favorable for vigor and growth are dominant, and genes that are harmful to the individual are recessive. It is true in the in our present knowledge also. That means when a gene becomes non-functional or due to mutation, it becomes as a mutant, right, or mutant trait. And the other, the functional gene will be dominant over the non-functional gene, so that when such alleles are brought together for different genes, all the dominant alleles will perform better, or all the dominant alleles will be brought together in the F1 hybrid, so that a hybrid vigor is observed. Then overdominance or codominance hypothesis was proposed by Shull and East in 1903 and 1908 respectively, and their findings were independent. According to this theory, the hybrid vigor on the base of heterozygosity is superior to homozygosity. That was the condition. According to this hypothesis, there are contrasting alleles for a character, A and A2, for a single locus. Each allele produces favorable yet different effects on the plant. That means there are two alleles of the same gene in the population. By hybridization, if they are brought together, both those alleles contribute to the character of the plant, so that it will be superior to the others. That means in codominance, the influence of both the alleles are taken into account, but in the case of theory of dominance, we take into account the effect of dominant allele only. That is the difference. In a heterozygous plant, A1, A2, for example, a combination of the effect is produced which is more favorable in the plant than the effect produced by either of the alleles, either A1 or A2, A2. A1, A2 will be superior due to their individual contribution to the character of the plant. The phenomenon of heterozygote, that means A1, A2, being superior to the homozygote, A1, A1 or A2, A2, is termed as overdominance. Various names for overdominance, superdominance, etc. were proposed, but it was finally the overdominance theory was accepted. So, what are the effects of heterosis? This is not actual effects. It is the most observed characters where heterosis plays a role. That is the meaning of effect here. That means which are the characters in which we can observe this heterosis. It is basically the result of increased metabolic activity that contribute to the heterosis. There may be quantitative effect and qualitative effect. In the case of quantitative effect, the quantitative characters are influenced, where the increase in size, increase in the length, height, etc. Hybrids are generally more vigorous, larger, healthier and faster growing than the parents. Head size in cabbage, guava, cob size in maize, fruit size in tomato, etc. are the examples where the size is increased, because size is a quantitative trait and multiple alleles come together, or many genes, these are polygenic traits, many genes at different locations, and the better alleles are brought together, it will get reflected in the phenotype of the organism F1. Then increase in yield. Yield may be measured in terms of grain, fruit, seed, leaf, tuber or the whole plant. Hybrids usually have increased yield. Then better quality. Hybrids show improved qualities, such as hybrids in onion show better keeping qualities. Then physiological effects are greater resistance to disease and pests, because all these alleles are brought together. It will surely show a wide range of resistance to pests and disease causing organisms. Then greater flowering and maturity. Earliness, it is the most important desirable trait in vegetables in many cases. Hybrids are earlier in flowering and maturity than the parents. That means tomato is an example where it will be early maturing. Greater adaptability. The hybrids can grow in wide range of environmental conditions. And the biological effects, where the hybrids exhibiting heterosis show an increased biological efficiency, that is an increase in the fertility, reproductive ability, etc. and the survival ability. So all these have some limitations. At the same time, if the hybridization is fully successful, we can expect such a increased response in the F1 progenies. Thank you.